All right, this is your lecture video for your chapter 9. Our sexual reproduction meiosis kind of goes and kind of wrapping up this unit on the functions of DNA and cells and cell cycle. So, starting off, when we did chapter 8, that was all about mitosis, which is asexual reproduction. That's what our normal body cells do, our somatic cells do to grow and repair. We do mitotic development. Some organisms only do mitosis. Bacteria only do mitosis to divide and to reproduce, but we use meiosis for reproduction. Our gametes, our sex cells, go through meiotic divisions to allow for reproduction, for fertilization to be able to occur. So this is called sexual reproduction. The DNA offspring comes from two different parents. You have each parent contributing half of the genetic material that's necessary, and when those cells come in, they combine and they restore that full that full genome. Okay? This is really helpful because it gives you diverse offspring. Meiosis is sexual reproduction is going to create diversity. Mitosis is asexual. There's really no diversity that comes with it, and that diversity is worth it. If there's an ecological disaster that's going to happen, if something occurs, a change in the climate, a change in the food availability, those organisms that are able to reproduce sexually combine with other organisms that are surviving, maximize their chances for survival, they're going to be able to continue along. And that probably does create some evolution. When you have two species that kind of encounter a divergent event, they start start to cause a divergent evolutionary tracks, which is how we get the rise between chimpanzees and bonobos and apes and those kind of things. They had common ancestors, but then they started to have different developmental tracks, most likely due to different ecological disasters or environmental conditions. If you don't have any genetic diversity, if you are a bacterium that specializes on a very specific molecule and that molecule runs out in the area that you're in, then you may not be able to adapt and continue, and your genetic line would stop. When we think about the human genome, there are 23 pairs of chromosomes. So 23 pairs means that there's 46 total chromosomes in all of your cells except for your sperm and egg. So we call that those pairs homologous pairs, and we'll kind of get into the definition of what really that is. But essentially, homologous pairs are going to be coding for the same genes, right? And so this might be a good time to review chapter 8 stuff and chapter 7, thinking about those definitions of genome, chromosome, gene, proteins, nucleotides, codons. If you're still not quite sure on that, it might be good to go back and kind of review the structures of our genome. We can actually look at our genome. So here you see an image of a karyotype. A karyotype is when they take out, they analyze your genetic material and they organize it based on the chromosome size. Each chromosome is numbered 1 through 23. That 23rd pair is the sex chromosomes, either XX or XY in this case. These are all arranged by both their size and their banding patterns. Banding patterns means that if you look at these chromosomes, you can see that there are different colors. Each of those colors is going to represent different sections, different genes on those chromosomes. And so based on the size, the banding patterns, the location of genes, and the centromeres that are holding the chromatids together, this protein right here in the middle that where you see it pinched together, all three of those things kind of help them match up chromosomes to each other. So right here you have chromosome number 1 in the beginning. It's the largest of them. You have one coming from one parent and the other coming from your other parent. And that's how it is for all 23 pairs. One from mom, one from dad. When they come together, they're going to each code their own proteins. They're each making proteins, which is why you kind of look like a mix between your parents usually. You kind of have a little bit of one, a little bit of the other. Maybe your eyes both maybe they have the size of your dad's, but then they kind of tilt like your mom's. And so the angle of them is slightly different. And so you have a combination because you have some proteins from each. And so you're going to be a combination of a lot of those traits. Sometimes the trait that you get from one parent might be recessive. So you really look like one parent. You have the same hair as your dad because your mom has the recessive trait for that hair color. And so you're getting your dad's dominant trait. So that could be why one parent is, I always think like the Weasley children from Harry Potter, right? You had the mom who was blonde or brunette, whatever she was, and then the dad was redhead, and all the kids were redhead. Well, that's not usually how it works. I don't know. I'm not great with hair genetics. That's pretty complicated. There's several genes with that. But that's that same idea that, well, if your one parent is passing down a recessive gene that doesn't make a protein, yeah, all the other children are going to have that one trait from the other parent because that is the dominant trait that's actually making a protein. That's just kind of trying to get you to connect this idea that every trait you have one from one from mom, one from dad, and those are going to determine what your trait actually is, what you actually have. When we look at this, when we talk about having two sets of chromosomes for each trait, for two sets of chromosome or a pair of chromosomes, you are called a diploid. Those are diploid cells, meaning di as in two. So for each chromosome, you have two copies. When we look at our sex cells, we're going to see those are haploid because they have half the number of chromosomes. But every cell in your body, except for your gametes, are going to have all 46 chromosomes, 23 pairs in every cell. The 1 through 22, these first 22 chromosomes are called your autosomes. They code for non-sex characteristics, hair color, height, the way you metabolize different proteins, the transport channels that are in your lungs, right? All of those different things, all those things that are very important and crucial to life. But again, not related to sexual development, non-sex characteristics. Those are your autosomes, those first 22. And typically they're about the equal size. Your autosomes are going to be the same size, same kind of banding patterns. When we look at our sex chromosomes, this last pair, the 23rd pair, if you're XX female, then they're pretty much the same size. But if you are XY, if you're male with XY, notice how small, you can kind of barely see it. You might be able to zoom in. That little fragment of a chromosome is the Y chromosome. It only has about 1,000 genes on it, and they almost all code for male characteristics. So they're all related to penis and testicle development. The X chromosome, you notice that males do still have an X chromosome. The X chromosome, hopefully that kind of triggers you then, oh, the X chromosome is more important than the Y. If you can survive with two X's or one X, you can still survive. You can't survive with just a Y chromosome. That would be a non-viable embryo. And so you have to have at least one X chromosome to be viable. You may have heard before that we all start off as kind of a female development. That's yes and no. Even in utero, we do start off developing along the same track, but then when you have that Y chromosome, after a few weeks, that Y chromosome kicks in and it starts to shift your development from female or from this common pathway towards the male pathway. Whereas if you don't have that Y, you continue along that, which is why if you get into any kind of when you get to reproductive anatomy and developmental anatomy, you'll see there are similar processes and similar tubes between male and female systems. And it's because we kind of have this similar chromosomal, we share these X chromosomes. So we said that kind of talked about this homologous pairs. Homologous sets, they carry the same genes. So again, one from mom, one from dad, the gene for eye color, for hair color, whatever it may be. But you're going to have one chromosome for each trait, or one chromosome from each parent. And so here you're seeing unreplicated chromosomes, right? So these are not sister chromatids. These are unreplicated. So you only have one chromosome. This is one, and this is one. They're different colors, and you can see that the genes are different. Here you have a capital A, capital B, little b on this one. And so it's in the same position, and it's the same centromere location, the same size. So they're all the same chromosomes. They're homologous chromosomes, but because they have different alleles, they have different variations of these genes, they're not identical. They're not sister chromatids. They're homologous pairs. And so that's something important to be able to understand, is that homologous pairs are going to be coding for the same genes, but they're going to have different alleles for those traits. And it's because your mom and dad are different. Hopefully they are not identical twins. That would be impossible, right? And so they have different variations of their genes, and that makes them homologous pairs. Again, that term is alleles. The different variations of a gene, of one specific gene, is the different allele for that trait. And so some traits have a couple of alleles, maybe one or two alleles. Some traits have 10 alleles, 10 variations. And so you can have a different combination of them. If you ever played that Kirby game, Crystal Shard, that's kind of what I think of, because Kirby you could take in one trait, or you could take in two traits. And if you took in two traits, they would combine to make something new. And then if you combine two different traits, that will make something different. And so that's kind of how I think about the combinations of alleles and how they pop up. So now here we have, we're seeing unreplicated versus replicated. So unreplicated, you have homologous pairs, but no sister chromatids. And the replicated, you have your homologous pairs, but each pair has now been duplicated to make it sister chromatid. So this is what happens during DNA replication. So these are, this would be an example of what they kind of look like during interphase, right? They're condensed in this way. So in interphase, right, they would be loose in their chromatin form, but it would be unreplicated. It would just be an unreplicated chromosome. During DNA replication, we go through that S phase of replicating our DNA, and this is what that means. Each homologous pair is going to make its sister chromatid. So remember, you don't have 4 chromosomes. You still only have 2 chromosomes, but now they're replicated, which allows them to divide. Each cell is going to be able to split apart, and it's going to be able to divide these sister chromatids so its daughter cells has the appropriate genetic material. It can be confusing to think about homologous pairs, so this book analogy is nice. Think about it as, sister chromatids would be the same editions of a book. Homologous pairs would be different editions of the same book. Edition 2 is going to be slightly different, maybe different text, maybe slightly different images, but it's still going to have the same kind of information. They're not going to completely rearrange the chapters. Chapter 4 is probably still going to be chapter 4, but it's going to have different information in it. And so that's kind of a way to think about homologous pairs versus sister chromatids. So with this chapter with meiosis, when we talked about how most of our cells are diploid cells, they have 2 copies of each chromosome. But for fertilization to occur during meiosis, after meiosis, or for fertilization to occur, your cells you can't have diploid cells fertilizing each other. Then you would end up with 4 copies of every chromosome. So for fertilization to occur, we have to have a haploid cell, which are gametes, sperm for males, eggs for females. And when you have 2 haploid cells coming together, oh yeah, now you end up with just 2 copies, a diploid cell. So this is where we kind of have this combination of mitosis and meiosis. So for organisms like eukaryotes, right, we are going to, let's start here with fertilization. So when fertilization occurs, you have the sperm and egg coming together, and they're creating a diploid organism. And once you create a diploid organism, now we're able to actually make proteins and grow and develop. After fertilization, it's all mitosis. The zygote goes through mitosis to grow in size. Growth and development do go hand in hand, and so it's going to develop into a juvenile and then to adult. And then once you hit adulthood, which technically is puberty for us, I know it's we do not think about people, 12-year-olds as adults, but technically that is they're hitting puberty around that age. Once you hit puberty, your eggs and your sperm start to develop. They start going through meiosis. So from birth to from fertilization until puberty, you are only doing mitotic development. It's once you get into puberty that you start getting signals to your sperm, to your testicles and ovaries, that it's time to go through meiosis and start producing gametes. You start producing eggs, start ovulating, you start ejaculating sperm, and then you are producing your gametes. You're producing your sex cells, which can combine to fertilize. And so you see how that process goes. We call these germ cells. Sex cells that are your gametes are your germ cells. We talk about the germ line versus the somatic cells on the somatic line, which are your normal body cells. So meiosis, what you're essentially starting with is a diploid cell, and it's going to go through 2 rounds of divisions. It uses the same division cycles as mitosis, so you've already learned those. We're just going to do them twice. The first round of division, we're going to be separating our homologous pairs, and then the second round of division, we separate the sister chromatids. So we start off with a diploid cell. After that first round, we've made haploid cells. So each of these 2 daughter cells has half the genetic material. We do 1 more round of divisions, and you create 4 daughter cells, 4 daughter haploid cells. So again, it's very similar to mitosis. The steps are the same: prophase, metaphase, anaphase, telophase, with cytokinesis in there. The difference is that, again, we're doing that twice. We still only replicate the DNA once because we're trying to make haploid cells. So we only replicate our DNA once. We divide it twice. We divide the nucleus twice. We also divide the cytoplasm twice. So cytokinesis and meiosis are going to happen 2 rounds. It's, again, very much the same. You start off with interphase, where your DNA is going to replicate G1, S, G2 phases. After that DNA replicates, it's going to go through those first rounds of prophase, metaphase, anaphase, and telophase. The difference here is that in meiosis, you're separating homologous pairs. So notice that instead of lining up all chromosomes along the equatorial plate in one line, they're stacked. You have a stack of homologous pairs. So that way, through this first round of divisions, you're going to end up still with 1 copy of each chromosome in a cell. Okay, it's not, each of these daughter cells is still going to have a chromosome 1, chromosome 2, chromosome 3. There's only 4 chromosomes. I'm sorry, there's only 2. or yeah, 4 chromosomes, 2n equals 4. There you go. The diploid number is 4. So there's only 4 chromosomes in this cell. And so each, for half the genetic material, 4 divided by 2 is 2. And so you want each daughter cell to have 2 chromosomes, 1, 2, 1, 2, 1, 2, 1, 2. The thing is, each of these chromosomes is going to be a homologous one to its neighbor. So this little one is chromosome, let's say this big one is 1, this little one is 2. This is chromosome 1, this is chromosome 2. This is chromosome 1, this is chromosome 2. This one's chromosome 1, this one's chromosome 2. So each daughter cell is still going to have one set for each chromosomes, but it's not going to have 2 sets. That's what we mean by haploid. So we're dividing our genetic material by 2. So that way it can combine with another cell and restore that ploidy, that number. So meiosis, reducing the number of chromosomes by half. The germ cell starts off with 2 pairs of homologous chromosomes. The gametes have 2 single unpaired chromosomes per cell. So we've divided the homologous pairs and the sister chromatids by the time we finish our meiosis. And again, it's just 2 rounds of that. The first round dividing homologous pairs, and then the second round we're dividing our sister chromatids. We're actually splitting the sister chromatids apart. This is helpful because meiosis is going to start creating genetic variability. By reducing the number of chromosomes to just a half amount, you allow it to recombine with other organisms. So fertilization is a way that we can increase genetic variability. And we're going to see a couple of the processes, the actual mechanisms within meiosis itself, within those divisions, that allow us to increase genetic variability. Crossing over and independent assortment are the other 2 big ones. So again, the process of meiosis is the same steps as mitosis. It's just that, remember, in meiosis 1, we're dividing homologous pairs. Meiosis 2, we're just separating sister chromatids. So during meiosis 1, prophase 1 of meiosis, there is an extra little step that happens, and it's because we're working with homologous pairs. We do this thing called crossing over, or our cells, our chromosomes, do this thing called crossing over. And this is where you have 2 homologous pairs, right? So this is chromosome number 1. So you have 1 from dad, 1 from mom. These 2 sister chromatids are actually going to kind of cross over. We call that place, it's the chiasma, where they're actually meeting at each other. They're going to kind of associate with each other there. And then what they're actually going to do is they're going to swap genetic material. They take a piece of chromosome from mom, a piece from dad, and they just exchange them only on that one sister chromatid, though. So notice what you have. You have this, look at this chromosome right here. So what was originally a sister chromatid, 2 sister chromatids attached to this chromosome, you now have 2 different chromatids. You have one that's the parental, that's the same as the original parent, and then one that's called a recombinant chromatid. We call it recombinant because it's recombined with its neighbor, with its homologous pair. So when we do crossing over, it occurs in prophase 1. Crossing over is going to occur between 1 sister chromatid from each homologous pair. So you end up creating 4 unique sister chromatids. And again, this is about increasing genetic variability. So now, instead of, even more so than randomly separating the homologous pairs, we've actually rearranged the genetic material. So you have 4 unique sister chromatids that are going to be in each of those gametes, those eventual sex cells. So it's just a way that, again, this idea that you have some traits that look like mom, some traits that look like dad. Oh, well, crossing over can help explain why that might be. You might, scramble that DNA a little more. The other one is the independent assortment. So this occurs in metaphase 1. So remember, in metaphase, you are lining up along the middle, and for meiosis, that's first round of meiosis, we're dividing homologous pairs. So the homologous pairs can line up either with, let's say you have all parent 1s on one side, parent 2 on the other, or we can swap one of those. Right? You could have 4 different alternatives. There's 3. This chromosome has this cell has 3 sets of chromosomes, so you can have 4 different alternatives here for how those chromosomes can line up. Again, notice that it's not swapping the actual chromosomes themselves. You can only swap your homologous pairs. You can't exchange chromosome 1 for chromosome 2, because then a daughter cell might end up with multiple copies of chromosome 2 and chromosome 1 and not have the other. And so you can only swap within the homologous pairs. You can only swap this way. You can't swap that way. But we can swap this way and alternate that division pattern. And so those are 2 big ways that we start to increase that genetic variability through our meiosis processes. Okay, so One exception for obviously our genetic individualism are twins. And so there are two kinds of twins. We call them monozygotic and dizygotic. Remember that word mono for one, zygotic for zygote. A zygote is what we call the cell right after fertilization, right when sperm and egg meet, that restores that genetic material. We call that a zygote. A zygote then goes through mitosis to create larger organisms. So you can be monozygotic, where you have one cell gets fertilized, one egg gets fertilized, and then that fertilized cell splits into two. Because you're starting with one fertilized egg with one zygote, these are going to have the exact same genetic material. Identical twins have the exact same genetic material as each other. Dizygotic twins, this is when you have two different eggs are fertilized by two separate sperm. That's why they are non-identical fraternal twins, because they have two different sets of genetic material. I have an aunt and uncle that are dizygotic twins. So you had two zygotes, two completely different zygotes being formed versus one zygote splitting into two. So that's that difference there. Twins aren't really an error. It's just a kind of a function of division. But there are actual errors that can occur. And so some of those can be harmless, but a lot of times for gamete development, when you start to have issues in splitting, you start to have some developmental characteristics or non-viable embryos created. Remember the splitting of chromosomes happens in anaphase. And after metaphase, anaphase, they start to split apart. So if they don't split apart properly, we call this non-disjunction. And so non-disjunction is this failure of either homologous pairs or sister chromatids to separate. So anaphase 1 or anaphase 2. You can see here these zygotes are going to end up with 3 copies, so an extra copy of each chromosome, and then these end up with 1 less. So you want them to have 2. We want our zygotes to have 2 copies, and this one has an extra and this one has fewer. So here you can see non-disjunction happening in anaphase 2. You end up with 2 normal zygotes, 2 zygotes with the correct number of chromosomes. This one has an extra chromosome, and then this one has 2 few. And so depending on what that chromosome is, it could be impactful or not. It could, and that degree of impact could vary, right? With these 2 zygotes, they're going to have a typical development. This extra one might have some developmental defects. You're probably already thinking about Down syndrome and the trisomy chromosomes. And so if you have an extra chromosome, you could still potentially survive, just depending on what that extra chromosome is and what it codes for. When you're missing a chromosome, though, again, it's much less likely for you to survive. The irony is the one that you can lose is that Y. If you don't have that Y chromosome, you can still survive. You can still develop, for the most part, normally or traditionally, however you want to say it, without being noticed. But if it's depending on which one it is, again, if it's that X chromosome, then you're not going to be viable. Like I was saying, Down syndrome is caused by non-disjunction, either in anaphase 1 or 2. I'm not really quite sure, but it might be either one. The most common one is having 3 copies of chromosome number 21, so trisomy 21. There are other kinds, though. I think 17 and 18, or chromosome 13 and chromosome 17, I believe, are common for non-disjunction. They tend to stick together, and so there are several trisomy disorders. But the one that we're most common with is this trisomy 21, where you have 3 copies of that 21st chromosome. Again, you can absolutely viable. There might be some developmental conditions, and those do vary. And so it's an interesting condition. I mentioned before that you can develop without your Y chromosome, but you have to have the X, to the point that you can have 3 X's, you can have 2 X's, and again, you can have just 1 X and still be perfectly viable and still be healthy. You might have you'll probably have some secondary sexual characteristics that are impeded, but for the most part, your development in your life can still proceed normally. So just a couple of these to kind of go through. There is the triple X syndrome, where you have an extra copy of an X. They tend to have a little bit taller stature, menstrual irregularities. That's a lot with the sexual non-disjunction disorders. They tend to impact exactly what you would think: menstruation, sexual reproduction, fertility issues. That tends to be where those conditions arise. If you are XXY, so if you have an X and a Y, you are male. That is a biological male. But having this extra X is really going to impede the secondary development. So you'll probably have smaller testicles, a smaller penis. You might have more fat deposits in certain areas, different kind of things. Because all people, male and female in between, we all produce testosterone and estrogen. If you have testicles, you produce a lot of testosterone. If you have ovaries, you produce a lot more estrogen. But your adrenal glands and your liver can both produce androgens, produce these sex hormones that can be converted at the cellular level. So if you have an XXY, there's potential for you to produce slightly more estrogen than someone that is just an XY. If you are, as we see in this one, XXX, you might have different levels of estrogen production, or this could impede your estrogen production. It just kind of depends sometimes on the individual and the condition. During early development, especially if you think about embryonic fetal development and for newborns, sexual characteristics can look pretty ambiguous in terms of the genitalia, a penis and a vagina for a truly newborn infant with a non-disjunction disorder. It can be pretty ambiguous. And up until the early 2000s, it was common practice for doctors to just in that moment go ahead and decide, oh, this is going to this will be easier for this child to develop as a female. So let's go ahead and start doing some procedures, start doing things to help them transition as a female, to go ahead and live as a female. In the last 20 years or so, that's practice has shifted to allowing children to develop and then make their decisions as to what fits them the best, depending on their development pattern. Because as you can imagine, for the way they did it previously, well, by the time you hit puberty, doctors weren't doing genetic testing when they were, on an infant to see, well, are you XXY? What's, the proper development here? They would just make a decision based on with the parents, what felt right at the moment. And then when you have a child go through puberty and those hormones start kicking in, oh, well, now what you were living as a female, well, your hormones are really producing a lot more testosterone. And so it can kind of have this very, very psychological jarring effect. And so best practice now through the American Psychological Association, American Medical Association, is to allow children to go through some stages of development to see what is going to be the easiest way for them to live, to exist. And so non-disjunction disorders are very interesting and somewhat topical. Other errors can occur besides the non-disjunction. You can have mutation, deletion, translocations. I'm not going to really, in terms of at least like exams and things, get big into having you identify specific mutation disorders. I'm just being familiar with that. There are several kinds. And again, that idea with mutations, that mutations can be beneficial, mutations can be harmful, or they can be just nothing. They can give no change at all. It just depends. However, typically when we're talking about moving sections of a chromosome, that's pretty big. You're swapping the locations of genes, and that can really start to mess with some stuff. We've talked about the stages of meiosis and how we have to create gametes for fertilization to occur. This next little section is how do we make gametes? You know that we do meiosis, but let's actually apply that to sperm and egg cells. Okay, so to make our germ cells, we have to create haploid cells. To make a haploid cell, you have to start with a diploid cell. Which maybe is a little backwards. Hopefully that makes sense in your brain. So sperm and egg cells start off as diploid cells, and we have stem cells. A stem cell is a cell that is able to continuously reproduce. They're kind of ever-living cells. So what happens is that you have here for sperm development, you start with what's called a spermatogonium. I always say with sperm and egg development, somebody had a lot of fun naming these. So you start off with what's called a spermatogonium. And a spermatogonium is a diploid stem cell. And one of the characteristics of stem cells is that once they divide, they'll produce an offspring, a daughter cell, but they also create another stem cell. So this spermatogonium divides through mitosis. It produces this daughter cell that's going to actually eventually become a sperm, but one of those daughter cells is going to be another spermatogonium that can go through another division, and that division will produce another spermatogonium. So stem cells, that's the characteristic of a stem cell, is that it's going to divide, but it's going to constantly be replicating itself so that it does not die, quote unquote. First round of spermatogenesis, spermatogonium is going to divide, create another diploid cell, or two diploid cells, rather. It's going to create two diploid cells. That's a mitotic division. The primary spermatocyte, this is then going to go through the 2 rounds of meiosis. And for sperm, you take one spermatocyte and you produce 4 actual mature sperm cells, 4 haploid cells, apologize. Start with 1 diploid, produce 4 haploid cells. Each of these can then go on to fertilize an egg potentially. Although eggs are only fertilized by 1 sperm. There's mechanisms to ensure that. For oogenesis, you start off with 1 oogonium, a diploid oogonium stem cell. Again, this fun words. Go ahead and say it with me. Oogonium. That oogonium is going to go through mitosis. It's going to produce its stem cell, its diploid stem cell, and a diploid primary oocyte. What's interesting with female development is that this oocyte is going to be frozen in development. It's going to be frozen in this stage. I'm sorry, it gets frozen at this this secondary oocyte. That's where it gets frozen. So primary oocyte, the diploid goes through 1 round of division for meiosis, and then during puberty, once we hit puberty, we go through another round. So this second round only happens after fertilization, after full development happens. Which is very interesting. What's also true is that you only produce 1 actual egg from a primary oocyte. So with male development, we have 1 spermatocyte produces 4 sperm. For females, you have 1 oocyte. It produces 4 daughter cells, 4 haploid daughter cells, but 3 of those are what we call polar bodies, and 1 of them is an ovum, is an actual egg that can be fertilized. This is just part of how the oocyte development works. It's just part of that process. Also, it's one of those things you don't want to be ovulating 4 eggs every month for potential fertilization. That would be quite a lot of children to bear at one time. And so just slightly different in those fertilization styles. All right. Thank you so much. Have a good day.